Good day, everyone. Welcome to Vanithi's weekly podcast on liquid biopsy, part 11, radiotherapy, circulating tumor cells, and radiosensitizers. Today is an inspirational and motivational podcast for future students who wish to contribute and pursue their careers in liquid biopsy research to save lives and improve cancer treatments. Let me start with my story of success on this topic, which attracted 425,000 Australian dollars research funding in a principal investigator role, which is now patented and in the process of licensing for commercialization with medonctherapeutics model in Perth as national and international competitive grants and awards winner of Sir Charles Gairdner Hospital and Curtin University postgraduate merit research scholarship with an additional 16 years of Indian government academic research excellency portfolio. Cellular radioresistance and radiosensitivity depend on cell type and origin, cell cycle, and genetic background. Deregulated pathways contribute to oncogenic cellular transformation, which manifests resistance to ionizing radiation. Altering gene expression will reverse the effect of radiation resistance therapy modalities. Focusing on identifying SFRP4 WNT inhibitor molecular targets responsible for inducing radiation sensitivity by using circulating tumor cells and cancer stem cells from cancer patient blood samples holds promise for liquid biopsy. This will advance the radiotherapy resistance mechanism for individualized therapy modalities in clinical applications. CTCs may possess features of the differentiated and heterogeneous population of stem-like tumor cells. Both epithelial and mesenchymal properties have been reported in breast, prostate, and lung cancers as cell clusters. The change in the CTC counts in response to radiation has a predictive diagnostic value for therapy response when compared to conventional tools. Regardless of the challenges and effort required to understand these rare cells biology, identifying CTCs will be a valuable addition to understand cancer metastasis. Studies reveal that CTCs from breast and prostate cancer can be cultured as spheroids or organoids for more than 6 months, and these CTCs possess self-renewal cancer stem cell properties and exhibit a radioresistant and chemoresistant phenotype with a high metastatic potential. Cancer stem cells and non-cancer stem cells possess common invasive migratory features such as epithelial mesenchymal transition. However, only cancer stem cells can give rise to metastasis. The overexpression of CD44 and CD133 cells provide tumor resistance and the capability for disease relapse after chemoradiotherapy in immunodeficient mice. Therefore, selective targeting of these markers should be utilized to deliver cytotoxic drugs to cancer stem cells. Research indicates radiotherapy makes the tumor cells more aggressive and potentiates the circulation of metastatic non-small lung cancer cells. In 2014, Olga Martin et al. Published an article in an international journal of radiation oncology biology physics investigating high CTC counts during early phase radiotherapy with gamma H2A foci DNA damage and repair mechanisms. Dorsey et al. 2015 reported telomerase activity in CTC by radiotherapy in non-small cell lung cancer patients and CTC salvage radiotherapy in prostate cancer patients. Our collaborative research group reported on the efficacy and tolerance of cisplatin in combination with radiotherapy in head and neck cancer patients, which has the potential to assess the influence of circulating tumor cells when using radiotherapy alone in head and neck cancer patients. DNA repair and replication candidate genes BRCA1, RAD51, FANCG, MCM7, CTC6, and ORC1 were found to be differentially regulated by post-radiation in prostate cancer cell lines. High human telomerase protein HTRT CTC count in localized non-small cell lung cancer showed the response of radiotherapy. Ionizing radiation-induced glioblastoma CTCs of head and neck cancer, prostate, breast cancer, and myeloid leukemia. To overcome radioresistance in colorectal cancer, a high level of nuclear beta-catenin accumulation was detected, showing promise for targeting beta-catenin as a therapeutic approach. Metastatic melanoma aggressiveness showed high expression of TRF2, SOX10, and CD10 in circulating tumor emboli. KEAP1, NRF2, and TP53 mutations in lung squamous cell carcinoma induced radiation resistance. To our knowledge, there are no papers published about the genetic modification of CTC to reduce their resistance and radiosensitivity as a treatment modality. Furthermore, investigating WNTs, CMET, PI3K, AKT, mTOR, cancer stem cells, epithelial markers, stem cells, and cancer stem cells marker in identifying circulating tumor cells will enhance radiosensitization modalities. Interesting to note that we cannot culture head and neck CTCs after chemoradiotherapy post-treatment using different methodologies. However, the early phase STEM CTC technology used in radiotherapy is warranted for SFRP4 circulating cancer stem cells and spheres predictions, clusters dissociation, and sensitization approaches. We are confident that the research will contribute to developing new therapeutic strategies for precision medicine. Thank you.